Very good morning to all the students. So, today we are going to discuss on the major chapter, that is sexual reproduction of flowering plants, which is from your NCERT 12. This is from your NCERT 12. So, yes, as yesterday how we have discussed about anatomy of flowering plants, today we are going to discuss pre-fertilization events, from sexual reproduction of flowering plants. As we all know, this is the very lengthy chapter, and every year definitely you can expect 3 to 4 questions in final NEET examination. Definitely 3 to 4 questions will come. So, yes, what are the concepts included in this chapter? Pre-fertilization events, under that you will learn microsporogenesis and megasporogenesis. Microsporogenesis, megasporogenesis, and in detail you will learn both about male gametophyte and female gametophyte of the angiosperm. And then after pre-fertilization events, you will be learning about, after pre-fertilization events, just a second. Yes. So, after pre-fertilization events, you will be learning about pollination, double fertilization, and then post-fertilization events on endosperm development, embryo development, ovule turning to seed, and ovary wall turning to fruit, and some special characteristic features or the concepts like at last apomixis and polyembryony. All these concepts we have to learn from sexual reproduction of flowering plants, which is a major chapter from biology NCERT 12 for your NEET examination. There is no chance or no way in skipping this chapter. Let us get started with the basic concepts from initial thing of the sexual reproduction of flowering plants. First, you have to know how flower is formed. Okay, how flower is formed. There is 3 to 4 lines in your NCERT that is given about the brief description on how the flower is formed. First, inflorescence is formed, that is shoot apical meristem turns to floral meristem. Shoot apical meristem will turn to floral meristem. We are talking only about angiosperms. Fine. So, what happens beta? Shoot apical meristem turns to floral meristem, and there are many hormonal and environmental changes that influence the change from shoot apical meristem to floral meristem, and this is done by a hypothetical hormone called as florigen. Okay, shoot apical meristem to floral meristem. Then this turns to inflorescence axis, and then this gives rise to floral primordia. So, clear beta? So, here what happens, how the flower is formed from floral primordia, a mature flower is formed. So, first what happens, shoot apical meristem to floral meristem by the hormone florigen. Floral meristem develops into an inflorescence axis by reducing the internodal length, and this turns to a pedicel, the stalk of the flower, and then this gives rise to a floral primordia that matures to a flower. In angiosperms of flower, you have 2 types of whorls. What do you have, ma? 2 types of whorls. You will call it as essential whorls and non-essential whorls. Essential whorls and non-essential whorls. Essential whorls include both androecium and gynoecium. Okay, essential whorls include what beta? Androecium and gynoecium. andrecium and gynoecium. Non-essential words include sepals and petals. Non-essential words include sepals and petals. So, this is the basic introductory part from sexual reproduction of flowering plants. You might be thinking that this is just an introduction, but many at times, one or two times, the questions are framed only from this flow chart, like assertion reason questions. In the most recent paper of 2025, you got an assertion reason question just from this framework. Okay, in 2023 also this is asked. So, nothing is introductory over here. This concept you have to learn. And what are the most questions that are framed from sexual reproduction of flowering plants include statement based questions, assertion reason questions, mostly you will get from this chapter. Okay. So, have keen interest on the complete concept, then only you will be in a position to answer statement based and assertion reason questions. So, now you have got an idea what are the essential words and what are the non-essential words. First, looking into the essential words, we will learn about the male reproductive structure. Male reproductive structure. Male reproductive structure includes, andrecium. You will also call it as stamens. Stamen includes a long stalk called as filament, and to the terminal end it has anther, bilobed anther. Fine. So, stamen includes long stalk filament and the terminal part of the filament has bilobed anther, and this filament is attached to the stalk. This filament is attached to the stalk, and this stalk we will call it as the proximal end of the filament is attached to the thalamus. The proximal end of the filament is attached to the thalamus. So, remember this term which is important, filament whose proximal end is attached to the thalamus and the distal end has bilobed anther. Clear, right? So, now if you see, this is about the male reproductive structure, that having long stalk and the proximal end is attached to the thalamus and the distal end is attached to a anther which is bilobed. If you have to look into the anther transverse section of anther, you can see anther is bilobed wherein it has two lobes. Okay, these are two lobes, lobe 1 and lobe 2. Okay, anther has two lobes and each lobe is having two theca, theca 1 and theca 2. Okay, so it is the male reproductive structure, that is stamen whose terminal part is having anther, terminal or distal part is having anther. This anther is bilobed and it has two lobes, each lobe is having two theca and it has the longitudinal dehiscence line. Okay, we will call it as line of dehiscence. Clear? So, now if you have to see clearly the transverse section of anther, you will analyze that anther has 4 layers, you will call it as 4 wall layers of anther, 4 anther wall layers also you can call it as anther wall layers. Listen carefully, very important. So, yes, what are the 4 anther wall layers? The outer layer is called as epidermis. Next to epidermis you have endothecium. Next to the endothecium you have tapetum. Okay, epidermis, endothecium and tapetum. At the center you have sporogenous tissue. So, all these layers, epidermis, endothecium, third layer is middle layers 2 to 3 and at the center you have tapetum. So you tell me now what are the 4 anther wall layers? In chat box you can type ma. First is what? Epidermis. Second is endothecium. Third, middle layers. Fourth is tapetum. Fine. So we have epidermis, endothecium, middle layers and tapetum. All the 3 layers, epidermis, endothecium and middle layers, they are protective in function. Okay, all these 3 are protective in function. They are protective in function. And epidermis is just single layered. It is made up of parenchymatous tissue. Note it down. It is made up of parenchymatous tissue. And next, coming to the endothecium. Endothecium has alpha cellulosic fibers. Alpha cellulosic fibers. This give hygroscopic nature, which help for the anther dehiscence. They help for anther dehiscence, and this is hygroscopic in nature. Help in anther dehiscence. Dehiscence is nothing but what beta? Release. Anther dehiscence. And next, middle layers can be 2 to 3. Okay, epidermis and endothecium are just one layer, but middle layers can be 2 to 3 layers. Middle layers can be 2 to 3 layers, and they are ephemeral. Okay, they are ephemeral. What is the term ephemeral means? They can degenerate after some period of time. Okay, so they are ephemeral. Last is tapetum. All these 3 layers are uninucleated. The cells in these 3 layers are uninucleated, but the last and innermost layer of the anther is binucleated or multi, or you can say this exhibits polyploid condition. Okay, tapetum exhibits polyploid condition. Okay, tapetum can be of 2 types beta. Okay, I am writing here, you see, tapetum can be of 2 types, glandular tapetum and secretory tapetum. Okay, glandular tapetum and secretory tapetum. Glandular tapetum will release out Ubish bodies. Ubish bodies which gives rise to sporopollenin. Okay, they give sporopollenin. They give sporopollenin. Just a second. So yes, they give sporopollenin, which is the composition of the outer pollen grain layer, outer wall layer of the pollen grain, which is made up of sporopollenin. Second is, this is also giving out the pollen kit. The composition which is present, the cellular constituents in the pollen grain are given by the tapetum wall only. So glandular tapetum is giving rise what? Ubish bodies that secretes sporopollenin, which is the outer wall layer of the pollen grain, and it has pollen kit. Just quickly remember all the wall layers or the structure of anther in detail beta. Focus just in 2 minutes. I will tell you the complete overview. The outer layer is what? Epidermis, which is parenchymatous. Next to that you have what? Endodermis. Endodermis have alpha cellulosic fibers which are hygroscopic in nature. Next to the endothecium you have middle layers which are 2 to 3 layered. They are ephemeral, means they will degenerate once the pollen grains are released. At the center you can see, sporogenous tissue. This sporogenous tissue will have microspore mother cells. Okay, sporogenous tissue will have microspore mother cell which undergoes meiosis, and they will give rise to haploid microspores. They will give rise to haploid microspores. This haploid microspores will mature to pollen grains which are haploid. They mature to haploid pollen grains. And once they mature to pollen grains, this middle layers which are ephemeral will be degenerated and endothecium will absorb all the water and cause dehydration of the pollen grains, and through the line of dehiscence the pollen grains will be released. So, yes, here you can see through the line of dehiscence the pollen grains are released. Fine. And here the tissue which is present in between the sporogenous tissue is called as connective tissue. So, this is all about the structure of anther. Clear beta? Now, we will go and clearly see the microsporogenesis, which is very important. So, in the time of microsporogenesis, as we have discussed, inside a sporogenous tissue, what is present? Microspore mother cells. What are present, ma? Microspore mother cells. This microspore mother cells are diploid. Microspore mother cells are diploid. Once this microspore mother cell is starting the division, we will call this microspore mother cell also as microsporocyte or meiocyte. Those cells which are ready to undergo meiosis, what do we call it as? Meiocyte. Remember, we will call it as meiocyte. Or as this is microspore mother cell, we are also calling it as microsporocyte. Okay? Microsporocyte. Remember all these synonyms, one and the same. Okay? These are diploid. Okay? This microsporocyte, which is diploid, will start undergoing meiosis. Okay? After the meiotic division, it gives rise to tetrad condition forming 4 haploid microspores. Okay? This gives rise to what? 4 haploid microspores, which will develop into pollen grain by grabbing all the nutrition which is given from the tapetum wall, which is the innermost wall layer. So, how the microspore is developed to pollen grain beta? Microspore is developed to pollen grain by haploid microspore. Remember, see it properly. Haploid microspore will mature to what? Haploid pollen grain. So, yes, Lakshita, good morning beta, good afternoon ma. Very good, you have given us all the wall layers of the anther. Focus on the class ma, you will have clear idea on complete pre-fertilization events and you will solve the NEET PYQs as well on this concept. Stay here for another 20 to 25 minutes, this concept will be very easy for you. So, haploid microspore maturing to haploid pollen grain by taking nutrition from which layer? Tapetal wall layer. Keep focusing on the class and keep responding ma. This tapetum, as we have discussed, it is of 2 types, right? Glandular tapetum and secretory tapetum. Glandular tapetum will release the sporopollenin and through Ubish bodies. This sporopollenin will form the outer wall layer of the pollen grain. The outer wall layer of the pollen grain, we call it as exine. Okay? The outer wall layer of the pollen grain is exine. This exine is made up of what ma? Sporopollenin. And there are few regions where you will call it as germ pore. Okay? Germ pore, wherein through this germ pore only, the pollen tube development starts once it reaches the stigma wall or stigma surface. Through the germ pore only the pollen tube growth starts. So, in the germ pore region of the exine, there is no sporopollenin. Remember that. Okay? So, the outer wall layer is made up of sporopollenin over which germ pores are there. That gives rise to pollen tube once the pollen grain lands on the stigma. That is why the germ pore region does not have sporopollenin. Next to the exine, you have intine wall. Next to this, what do you have ma? Intine wall. Intine wall is made up of cellulose and pectin. Okay, intine wall is made up of cellulose and pectin. Remember the compositions, ma, that is very important, cellulose and pectin. Fine. So, here you can clearly see outer wall, that is exine, inner wall, that is intine, and all the cellular constituents whichever are present inside the pollen grain are given from the secretory tapetum. Okay, they are built up by the secretory tapetum. So, yes, this is what the pollen grain structure is. In most of the angiosperms, like in 60% of the angiosperms, the pollen grain is released in two-celled stage. Pollen grain is released in what stage, beta? Two-celled stage. So, what is happening over here? Microspore mother cell which is diploid undergoing meiosis forms haploid microspores. They get mature to what? Pollen grains. This pollen grains undergoes one mitotic division forming two cells inside it. You will call those two cells as vegetative cell and generative cell. You will call it as what, Nana? Vegetative cell and generative cell. So, how you can vegetative cell and generative cell? Mostly in this two-celled condition only the pollen grain is released. How you can identify between the vegetative cell and generative cell? Firstly, there is large vacuole formation inside the vegetative cell and abnormal shaped nucleus. Vegetative cell has abnormal shaped nucleus. Excuse me. And generative cell has spindle shaped nucleus. Generative cell has spindle shaped nucleus. This is all about the pollen grains. Clear, ma? The structure of the pollen grain and the two-celled stage of release which has vegetative cell and generative cell. Generative cell has spindle shaped nucleus, vegetative cell has abnormal shaped nucleus. That is it. Now we will understand the release of the pollen grains, that all we know. Now we will clearly understand the female gametophyte structure also, and then we will go for pollination processes and all. So, female gametophyte in angiosperms is gynecium. And what makes one carpel? Okay, one carpel. One carpel includes one unit of, okay, it includes one unit of stigma, style and ovary. Okay, it includes what, beta? One unit of stigma, style and ovary. Fine. So, one unit of stigma, style and ovary is making you one carpel, is giving you one carpel. As such, many units of carpel will give rise to multicarpellary condition. Whether it is one carpel or more than one carpel, you will call it as what? Pistil. One carpel or more than one carpel, you will name it as pistil. So, carpel and pistil you will use as synonyms, but do remember one carpel means it is having one unit of stigma, style and ovary, and then you will call it as monocarpellary or single pistil. Monocarpellary or single pistil. For example, examples under monocarpellary condition will have mango, coconut and pea. Multicarpellary condition where it has more than one pistil, either they can be united like this or the carpel units can be free like this. Okay, the carpel units can be free like this. If they are united, you will call it as syncarpus where pistils are fused. Syncarpus where pistils are fused. For example, papaver and hibiscus. Okay, just a second. Okay, no issues. So, yes, we have learnt carpels can be of two types, right, ma? Carpels can be of two types. Those can be monocarpellary and multicarpellary, right? Multicarpellary can be apocarpus and syncarpus, right ma'am? If it is syncarpus, the examples include like papaver and hibiscus. Do remember, papaver and hibiscus. You can remember this also like HP, papaver and hibiscus, or hibiscus and papaver, wherein all the pistils are fused. Second type is apocarpus, wherein carpels are free. It is multicarpellary and free, you will call it as apocarpus. Best example is Michaelia. Fine, understood? This is multicarpellary condition. Monocarpellary, where it has single carpel, that is single unit of stigma, style and ovary. Example, best example includes mango. Fine, these are about the structure of gynoecium. Now if you see stigma, you know this is the receptive site of the pollen grain. Style, the long tube like structure which has pollen tube in it after fertilization. Before fertilization, once the pollen grain lands on the stigma, that develops the pollen tube. This you all know, right? Now inside ovary, you look carefully. Inside ovary there is complete fluid filled space, you will call it as placenta. Like how in human mother there is mother's womb, there is complete placenta. In the same way, inside ovary there is placenta, and inside placenta you will have locules like this. Okay, inside placenta what do you have, ma? Locules, and in this locules ovules are placed. In this locules ovules are placed. The arrangements of ovule inside the ovary is called as what? Placentation. Okay, it is called as placentation. As such, we have learnt marginal placentation, axial, parietal and free central, basal placentation in morphology of flowering plants. Do remember what is happening here? Inside ovary there is complete fluid filled space called as placenta, which has locules. Inside the locules ovules are arranged in any way. That mode of arrangement of ovules inside the ovary is called as placenta. It is of 5 types: marginal, axial, parietal, free central and basal placentation. Now if you have to see, ovule arrangement is different in different plants. Okay, we have different types of ovules: orthotropous, anatropous, hemianatropous ovules. We have many types of ovules as such. But in most of the angiosperms or in flowering plants, the type of ovule which is present is inverted ovule. Inverted ovule. Okay, inverted ovule which is present in most of the angiosperms. So here if you see ovule parts, labelings of the ovule which are very important, we can expect diagram based question from here also. So the stalk of the ovule through which the ovule can attach to the placenta of the ovary, that stalk of the ovule is called as funicle. So what is funicle, beta? Funicle is nothing but stalk of the ovary. Stalk of the ovule, sorry. Okay, funicle is the stalk of the ovule. And there is small structure which is attaching funicle to the ovule. This structure is called as hilum. Okay, hilum. Post fertilization, when ovule matures to seed, hilum looks like a scar on the seed. Hilum looks like what? Scar on the seed post fertilization. Okay, just a second. Lakshita, it is stalk of the flower. Good ma, keep responding and analyze the class properly. So yes, funicle or hilum in the inverted ovule will be close to the pore called as micropyle. Okay, this is close to the pore called as what? Micropyle, and opposite to the micropyle you have what? Chalaza. Okay, chalaza is the base of the ovule. Okay, and ovule is covered or surrounded by what? Integuments. Cells inside the ovule is called as nucellus. Cells inside the ovule are called as nucellus. So, till here everything is clear now, parts of the ovule. Once let me recall you once again. Stalk of the ovule is what? Funicle, and the small structure which is attaching funicle to the ovule, this is called as hilum. Hilum post fertilization, this appears on the seed like how? Scar on the seed. Fine. And close to this what is present? Micropylar end. Opposite to this chalazial end, which is the base of the ovule. And this ovule is surrounded by integuments. It can be unitegmic or bitegmic or tritegmic. This integuments in the ovule, post fertilization, they will form the seed coat in the seed. Do remember. Now, inside ovule what cells are there? Nucellus cells are there. What is the ploidy of nucellus cells? Important, that is diploid. Okay. Nucellus cells ploidy is what ma? Diploid. Remember. So, now if you see megasporogenesis, which is very important question, recently asked in 2025 also, please have focus here. So, here we all know the cells present inside the ovule are called as nucellus. Those nucellus cell, one of the nucellus cell which is close to the micropylar end will differentiate into megaspore mother cell. Okay, it differentiates to megaspore mother cell. Fine. So, this megaspore mother cell is diploid. This cell which is close to what end ma? Micropylar end. Remember, micropylar end. So, here one of the nucellus cell which is close to the micropylar end is differentiating to megaspore mother cell. This megaspore mother cell undergoes meiotic division. Meiotic division, you will also call it as what? Reductional division. And this forms a linear tetrad like this. Okay, linear tetrad. Fine. Let's suppose this is the chalazial end and this is the micropylar end. Of these 4 cells, 4 daughter cells, these are called as haploid megaspores. Megaspores. Do remember. Okay. So, megaspore mother cell which is close to the micropylar end, undergoing one meiotic division, it forms 4 haploid megaspores. Okay, 4 haploid megaspores, micropylar and chalazial end. Remember, of this 3 haploid megaspores which are at the micropylar end will degenerate. Okay, this will degenerate. Fine. And one cell which is close to the chalazial end is functional. Remember that. Which is the functional megaspore? Functional megaspore is at chalazial end. Fine. Clear, right? So, you have 3 cells which are at the micropylar end, they will degenerate, and one cell which is at the chalazial end, that is functional megaspore. Now, this functional haploid megaspore will undergo 3 sequential mitotic divisions beta. How many divisions? Here you can see one functional megaspore, this one functional megaspore which is at what end? Chalazial end. Undergoes how many divisions? No worries, I'll repeat once again about megasporogenesis, but now analyze the process properly. One functional megaspore which is at the chalazial end will undergo 3 sequential mitotic divisions. After first mitosis, okay, mitosis 1, 2 nuclei are formed, mitosis 2, just a second, mitosis. How many mitosis? 3 mitotic divisions. First mitotic division, 2 nuclei are formed. Second, 4 are formed. Third, 8 nucleate condition is formed. Of course, 3 mitosis is happening, but there is no cytokinesis until 3 mitotic division completes. After third mitotic division, if you see, you can clearly look into 8 nucleated 7 celled condition, which is the embryo sac. What is that, Nana? 8 nucleated 7 celled condition. Fine. So, this is 8 nucleated 7 celled condition. In this 8 nucleated, okay, at the chelical end 3 cells are present. These 3 cells are called as antipodals, and at the micropylar end you can see 2 cells, you call it as synergids, and 1 cell attached to it is what? Egg cell. At the center 2 nuclei which form polar nuclei. Okay, this is polar nuclei, or this is central cell which is of diploid condition. Whereas complete cells in the embryo sac are haploid. So, embryo sac is haploid. Embryo sac is haploid. What is the female gametophyte in the angiosperms? Embryo sac, which is the female gametophyte in the angiosperms. Female gametophyte. Why is it called as female gametophyte, ma? Because it is bearing the female gamete, that is egg. That is why it is called as female gametophyte. Once again, I will quickly repeat you the complete process of megasporogenesis. Listen carefully. So, from the structure of the ovule, it is very clear that inside ovule what cells are present? Nucellus cells are present. These nucellus cells are diploid. This diploid nucellus cells, one of the nucellus cell which is close to the micropylar end will differentiate into megaspore mother cell, which undergoes meiotic division or reductional division, forming 4 haploid megaspores, which is a linear tetrad. 3 megaspores, 3 haploid megaspores of micropylar end will degenerate, and 1 haploid megaspore towards the chelical end will be functional megaspore. This megaspore, which is functional, will undergo 3 sequential mitotic divisions. After first division, that forms 2 nucleate condition, second, 4, third and 8 nucleated condition are formed. At the end, if you see, at the micropylar end, synergids are there and egg cell. Synergids and egg cell together you will call it as egg apparatus. Okay, synergids and egg cell in the micropylar end together you will call it as what, ma? Egg apparatus. And at the center you have central cell which is having 2 polar nuclei, and at the chelical end you have 3 antipodal cells which are haploid. So, embryo sac is what? Haploid female gametophyte in the angiosperms. Now, it is clear for you know how embryo sac is formed. At the micropylar end you have 2 synergids. Synergids will be having what? Filiform apparatus, and egg is attached to this. Egg and synergids together you will call it as egg apparatus, and here is the central cell and antipodals towards the chelical end. So, now whenever the receptive pollen grain lands on the stigma, just look at the structure. Fine. So, here an embryo sac. So, this embryo sac, just a second. Yes. So, inside ovule, after 1 meiotic division and how many mitotic divisions, ma? After 1 meiotic division and 3 mitotic divisions, 3 sequential mitotic divisions, what cells are formed? At the chelical end you have 3 antipodal cells, this is the chelical end, and at the micropylar end you have egg and 2 synergids, okay, egg and 2 synergids with filiform apparatus, and at the center you have a central cell. This is the micropylar end. Whenever the pollen grain which is receptive is landing on the stigma, okay, pollen grain which is receptive is landing on the stigma, we have learned, right, the exine wall is made up of what? Sporopollenin. Those regions where sporopollenin is not there, from there pollen tube will germinate, okay, pollen tube will germinate, and here intine wall will be there, inside that you have vegetative cell and generative cell. Generative cell, when it is travelling inside the pollen tube only, it will give rise to 2 male gametes. Okay, it will give rise to what ma? 2 male gametes. These 2 male gametes, once they land the micropylar end, yes, one male gamete will fuse with egg cell, other male gamete will fuse with polar nuclei. So, this male gamete fusing with egg cell is called as syngamy. This is called as what ma? Syngamy. And one more male gamete fusing with the central cell or polar nuclei is called as triple fusion. Okay, this is called as what? Triple fusion, which gives rise to primary endospermic nucleus. This gives rise to what? Primary endospermic nucleus. And as 2 fusions happen, 2 fertilizations are happening, you will call this fertilization as double fertilization in angiosperms. This is what ma? Double fertilization. So, I hope all the pre-fertilization events are clear to you, microsporogenesis, megasporogenesis and the gist of double fertilization. From this concept, we are going to solve the previous year questions 2023 to 2025. Stay focused and answer better. So, now if you see assertion reason question which is asked in the year 2023, wherein assertion, a flower is defined as modified shoot. Initially from the introduction part we have learned, right. So, a flower which is called as PN, Lakshita, PN is called as polar nuclei beta. PN is called as polar nuclei. Clear ma? Yes. So, now try to answer these questions beta. First, assertion, a flower is defined as modified shoot, wherein shoot apical meristem is turned to or changes to floral meristem. Assertion is true only. Internode of the shoot will get condensed to produce different floral appendages laterally at successive nodes instead of leaves. Instead of leaves, what happens? Internodal length will be reduced and they will give rise to different floral appendages. So, reason is also true and reason is explaining assertion. So, both A and R are true and reason is clearly explaining the assertion how floral meristem is turning, how shoot apical meristem is modified to floral meristem is clearly explained in the reason. So, we have to go with the option 1. Next, in 2024, this is about embryo sac. In angiosperms, the haploid, diploid, triploid structures of fertilized embryo sac. He is asking about the fertilized embryo sac. Just now we have seen. First is the haploid structure of the embryo sac. Synergids are haploid, true. Antipodals haploid, synergids are haploid, true. All these beginning cells are haploid only. Diploid, primary endospermic nucleus is triploid, it is not diploid. So, you can eliminate this option. Synergids, they are haploid, so you can eliminate this also. This zygote, which is the primary product after fertilization, this is diploid. So, you can go with this answer. Next, triploid is what ma? Primary endospermic nucleus. So, Lakshita, tell me the answer beta. It is very simple, no? Option 3. Good. Next, answer this question ma, 2025. Again assertion reason question. That is why I told you from sexual reproduction of flowering plants you will get more of statement based and assertion reason questions only. So, here if you see a typical unfertilized angiosperm embryo sac at maturity, this gives what? 8 nucleated 7 celled condition, correct. Next, the egg apparatus has 2 polar nuclei, wrong. Just now we have seen, right, egg apparatus is having what ma? Egg apparatus will have one egg cell plus 2 synergids, 1 egg cell and 2 synergids. That is it. So, yes, here what answer is right to go through? Assertion is true and reason is false. Next question. So, here if you see again an assertion reason question in the year 2025. Cells of tapetum, this is from the anther wall layer, okay, this is from anther wall layer. So, here if you see, cells of tapetum possess dense cytoplasm, generally have more than one nucleus. True? Tapetum is having what? Polyploid condition. Next, presence of more than one nucleus in the tapetum increases the efficiency of nourishing developmental microspore mother cell. Reason is true, this is nourishing the developmental. Once again you see carefully, cells of the tapetum possess dense cytoplasm and generally having more than one nucleus, this is true. Second, the presence of more than one nucleus in tapetum increases the efficiency of nourishing development microspore mother cell. True. Reason is explaining assertion. Why it is the multinucleated condition is being explained. That it is multinucleated or exhibiting polyploid condition to provide nutrients to microspore mother cell. Next you see again, you got 3 questions in 2025 recent NEET paper. How many meiotic and mitotic divisions need to occur for the development of mature female gametophyte? That is what? Embryo sac. Embryo sac from megaspore mother cell in an angiosperm. How many meiotic and mitotic divisions have to happen, ma? 1 meiotic and 3 mitotic divisions. Option 1 will be correct. Last, so yes, with this we have practiced all the questions from 2023 to 2025 and we have made clear in this class about pre-fertilization events and in brief about double fertilization events. Coming class we will build up one more strong bridge course session. Stay tuned and thank you. Very good Lakshita. Join for the next session as well beta. Thank you.